In 1969, Robert Harding Whittaker introduced a revolutionary system for classifying all living organisms. Before Whittaker, biological classification was primarily based on a two-kingdom system, dividing life between plants and animals. This system failed to account for microorganisms, fungi, and many unique life forms that didn't fit neatly into the plant or animal categories. Whittaker expanded biological classification to 5 distinct kingdoms: Monera, Protista, Fungi, Plantae, and Animalia. Whittaker's system was based on 3 key criteria: cell structure, body organization, and mode of nutrition. Each kingdom is classified according to distinct characteristics of cell structure, organizational complexity, and nutritional strategy. Monera consists of prokaryotic unicellular organisms like bacteria. Protista includes mostly unicellular eukaryotes. Fungi are primarily decomposers with a unique absorptive nutrition. Plantae are multicellular photosynthetic organisms. And Animalia includes heterotrophic, multicellular organisms that ingest their food. Whittaker's classification system represented a significant advancement in biological taxonomy. It recognized the diversity of microorganisms, established fungi as a separate kingdom, and laid the groundwork for our modern understanding of evolutionary relationships. While later advancements in molecular biology would further refine our understanding of biological relationships, Whittaker's 5-kingdom classification remains a foundational framework that revolutionized how we organize and understand the diversity of life. The history of biological classification spans over 2,000 years, evolving as our understanding of life expanded. Aristotle developed one of the earliest classification systems around 350 BCE. He categorized organisms primarily by their habitat, as land dwellers, water dwellers, or air dwellers. This simple system was based on observation but had significant limitations. For example, whales were grouped with fish rather than mammals. In 1735, Carl Linnaeus introduced a more systematic approach with his 2-kingdom classification, Plantae and Animalia. Plants were characterized as immobile organisms that produced their own food, while animals were mobile organisms that consumed food and possessed sensory organs. However, as scientific knowledge expanded, particularly with the invention of the microscope, many organisms were discovered that didn't fit neatly into either kingdom. In 1866, Ernst Haeckel proposed a 3-kingdom system that added a new kingdom called Protista to accommodate single-celled organisms like bacteria, protozoa, and algae. This was a significant improvement, but still had limitations. For instance, fungi were still classified with plants despite their distinct characteristics. In 1969, Robert Whittaker proposed the 5-kingdom classification system that became widely accepted for decades. Whittaker based his classification on 3 main criteria: cell type, cellular organization, and mode of nutrition. This system became widely accepted because it reflected evolutionary relationships while making practical distinctions between major groups of organisms. The journey from Aristotle to Whittaker demonstrates how classification systems evolved to incorporate new scientific knowledge, moving from simple observation-based systems to those based on cellular and genetic relationships. Kingdom Monera consists of all prokaryotic organisms, including bacteria and archaea. These are single-celled organisms without a true nucleus or membrane-bound organelles. Unlike other cells in other kingdoms, prokaryotic cells have a simple structure. Their cell walls typically contain peptidoglycan, which gives them structural rigidity. Instead of a true nucleus, they have a nucleoid region where their DNA is located. Their DNA is circular, unlike the linear chromosomes found in eukaryotes. Ribosomes are present for protein synthesis, but they are not attached to any membrane systems. Many bacteria have flagella for movement, allowing them to move toward nutrients or away from harmful substances. Monerans exhibit diverse nutritional modes, allowing them to thrive in virtually every environment on Earth. Photosynthetic bacteria, like cyanobacteria, use light energy to produce their own food. similar to plants. Chemosynthetic bacteria derive energy from inorganic chemical reactions, often in extreme environments like deep-sea vents. Heterotrophic bacteria, such as E. coli, obtain energy by consuming organic matter from their surroundings. Let's explore some representative examples of kingdom Monera. Cyanobacteria, formerly known as blue-green algae, are photosynthetic bacteria that produce oxygen and are essential to Earth's early atmosphere development. Escherichia coli, or E. coli, are common bacteria found in the intestines of humans and animals. While some strains are harmful, most are beneficial. Archaebacteria represent an ancient lineage of prokaryotes that often inhabit extreme environments, such as hot springs, salt lakes, and deep-sea vents. Monerans play crucial ecological roles in every ecosystem on Earth. As decomposers, bacteria break down dead organic matter and recycle nutrients, making them available for other organisms. Certain bacteria can fix atmospheric nitrogen into forms that plants can use, which is essential for plant growth and agriculture. In summary, Kingdom Monera comprises prokaryotic organisms with a relatively simple cellular structure, but they display incredible diversity in their metabolic abilities and ecological roles. Kingdom Protista comprises a diverse group of primarily unicellular eukaryotic organisms. Protists have several key features that distinguish them. First, they possess a membrane-bound nucleus, making them eukaryotes. They contain specialized organelles like mitochondria for energy production, and in some species, chloroplasts for photosynthesis. Protists exhibit various modes of nutrition. Some are autotrophic, making their own food through photosynthesis. Others are heterotrophic, consuming other organisms. Some are even mixotrophic, using both methods. Protists are incredibly diverse and are often categorized based on their characteristics and nutritional methods. Algae are photosynthetic protists commonly found in aquatic environments. They can be unicellular or multicellular and often contain chlorophyll. Protozoa are animal-like heterotrophic protists. These include amoebas, paramecia, and other mobile single-celled organisms. Slime molds have characteristics of both fungi and amoebas. They can move and feed like animals, but reproduce with spores like fungi. Water molds are absorptive, fungus-like aquatic organisms. Despite their name and appearance, they're not true fungi, but protists. Unlike other kingdoms with clear defining characteristics, Protista was often considered a somewhat artificial grouping. It essentially became a catch-all kingdom for eukaryotic organisms that didn't clearly belong in Plantae, Animalia, or fungi. For example, algae share characteristics with plants, but are different enough to not be classified as plants. Similarly, protozoa have animal-like traits but aren't animals, and slime molds resemble fungi in some ways but have unique protist features. In summary, Kingdom Protista represents a diverse collection of eukaryotic organisms with varied characteristics, serving as an evolutionary bridge between simpler prokaryotes and more complex multicellular organisms. Kingdom fungi are a diverse group of organisms that play a vital role in our ecosystems as decomposers. Fungi are eukaryotic organisms with cells that contain a membrane-bound nucleus and organelles. Unlike plants, fungi cell walls contain chitin rather than cellulose, which gives them structural rigidity. The fungal body consists of thread-like structures called hyphae. These hyphae form an interconnected network called mycelium, which spreads through the substrate to absorb nutrients. Fungi are heterotrophs that obtain nutrients through absorption, unlike animals which ingest their food. They release digestive enzymes into their environment to break down complex organic materials externally. The broken down nutrients are then absorbed through the fungal hyphae, allowing them to obtain energy and building materials. Fungi reproduce primarily through the production of spores, which are specialized reproductive cells. These spores are lightweight and can be dispersed by wind, water, or carried by animals to new locations where they can develop into new fungi. Many fungi can also reproduce asexually through fragmentation of their mycelium or through budding, as seen in yeasts. The kingdom fungi includes over 144,000 known species, with many more yet to be discovered. Some familiar examples include mushrooms, the reproductive structures of many fungi that we can see above ground. Yeasts are single-celled fungi that are essential in baking and brewing. Molds are multicellular fungi that grow in filaments and are often found on food and in damp environments. Fungi serve as the primary decomposers in most ecosystems, breaking down complex organic materials like cellulose and lignin that few other organisms can digest. Through decomposition, fungi recycle nutrients locked in dead organisms and return them to the soil, making them available for other living things. Many fungi also form important symbiotic relationships with plants as mycorrhizae, helping plants absorb nutrients, or with algae as lichens. Fungi have significant economic importance in various industries. In food production, they're used to create mushrooms, cheese, bread, beer, and wine. In medicine, fungi are the source of many important drugs, including antibiotics like penicillin, cholesterol-lowering drugs, and immunosuppressants used during organ transplants. In biotechnology, fungi provide enzymes used in detergents, biofuel production, and bioremediation of environmental pollutants. Kingdom Animalia comprises multicellular eukaryotic organisms that lack cell walls and consume other organisms for nutrition. Animals have several distinctive characteristics that set them apart from other kingdoms. First, animals are multicellular organisms with eukaryotic cells containing membrane-bound organelles and a true nucleus. Unlike plant cells, animal cells lack rigid cell walls, allowing for greater flexibility and mobility. Animals are heterotrophs, meaning they obtain energy by consuming other organisms, a process called ingestion. They possess complex tissue organization with specialized cell types arranged into functional layers. These tissues form specialized organs and organ systems that perform specific functions. While some animals can reproduce asexually, most rely on sexual reproduction, combining genetic material from two parents. Animal cells have a distinctive structure. They possess a flexible cell membrane, but lack the rigid cell wall found in plant cells. Like other eukaryotes, animal cells contain a nucleus that houses genetic material and various organelles like mitochondria that perform specialized functions. Animals are heterotrophs, obtaining nutrients by consuming and digesting other organisms. The digestive system is specialized for processing food. The process begins with ingestion through the mouth. Food passes through the digestive tract where it's physically and chemically broken down in the stomach. Finally, nutrients are absorbed in the intestines and waste products are eliminated from the body. Animals have complex tissue organization, with cells arranged into specialized functional layers. 4 primary tissue types are found in animals. Epithelial tissue forms protective barriers, connective tissue provides structural support, muscle tissue enables movement, and nervous tissue transmits signals. These tissues combine to form specialized organs and organ systems that perform specific functions in the animal body. Animals exhibit remarkable diversity, ranging from simple organisms to highly complex forms. The simplest animals are sponges, belonging to phylum Porifera. They lack true tissues and organs, but have specialized cells. Cnidarians, like jellyfish, have radial symmetry and tentacles with stinging cells. Arthropods, including insects and crustaceans, have segmented bodies, exoskeletons, and jointed appendages. They are the most diverse animal group. Vertebrates, such as fish, amphibians, reptiles, birds, and mammals, have a backbone and the most complex organ systems. Animals have evolved diverse adaptations for movement, sensing their environment, and surviving in various habitats. Movement adaptations include fins and tails for swimming, wings for flying, and specialized limbs for running or crawling. Sensory adaptations allow animals to perceive their environment with specialized structures for vision, hearing, smell, and touch. Environmental adaptations help animals survive in specific habitats, including camouflage, thermoregulation mechanisms, water conservation, and seasonal behaviors like hibernation or migration. Whittaker's 5-kingdom classification system offers several significant advantages over previous systems. First, it recognizes the fundamental differences between prokaryotes and eukaryotes. This distinction acknowledges the major evolutionary leap from simple cells to complex ones with membrane-bound organelles. Second, it acknowledges fungi as a distinct kingdom separate from plants. Previous systems classified fungi as plants, but Whittaker recognized their unique characteristics, including their absorptive nutrition and chitin cell walls. Third, Whittaker's system uses multiple criteria for classification, including cell structure, body organization, and mode of nutrition. This multi-factor approach leads to more accurate and natural groupings of organisms. Fourth, Whittaker's system better reflects natural evolutionary relationships between organisms than previous systems. By considering fundamental biological characteristics, it groups organisms that share common evolutionary history. Finally, Whittaker's system provides practical utility for organizing biological diversity in a comprehensible framework. This organization facilitates both education and research by creating logical groupings that help scientists and students understand relationships between organisms. In summary, Whittaker's five-kingdom classification system offered significant improvements over previous systems by recognizing fundamental biological differences, using multiple criteria, and providing a practical framework that better reflected evolutionary relationships. A major limitation of Whittaker's classification system is its difficulty handling organisms that share characteristics of multiple kingdoms. Molecular data has revealed evolutionary relationships that contradict Whittaker's system. For example, fungi are actually more closely related to animals than to plants, which wasn't recognized in the Five Kingdoms system. Kingdom Protista became a problematic catch-all category. It artificially grouped together diverse organisms like algae, protozoa, slime molds, and water molds that have very different evolutionary origins. Whittaker's system couldn't properly place viruses, which exist at the boundary between living and non-living things. Viruses lack cellular structure and independent metabolism, yet they contain genetic material and can reproduce within host cells. Whittaker's classification doesn't account for endosymbiotic relationships. Modern evidence shows that mitochondria and chloroplasts originated as free-living bacteria that were engulfed by primitive cells, creating complex evolutionary relationships not reflected in the five-kingdom system. These limitations led Carl Woese to propose the three-domain system in 1977. Based on genetic analysis of ribosomal RNA, Woese separated prokaryotes into two domains, bacteria and archaea, while placing all eukaryotes in domain eukarya. This system better reflects evolutionary relationships revealed by molecular data. Modern classification systems have evolved dramatically with advances in scientific knowledge. The most significant change has been the use of molecular and genetic data for taxonomy. DNA sequencing, protein comparison, and gene expression analysis provide deeper insights into evolutionary relationships. These molecular insights led to the three-domain system proposed by Carl Woese, which divides life into bacteria, archaea, and eukarya. This system recognized that some prokaryotes, the archaea, are as different from bacteria as they are from eukaryotes. Modern classification also employs cladistics, which uses shared derived characteristics to create phylogenetic trees that represent evolutionary relationships. These trees show how species diverged from common ancestors, providing a more accurate picture of biodiversity. It's important to recognize that classification systems are scientific models that change as our understanding improves. While Whittaker's five-kingdom system has been largely superseded, it remains historically important and continues to be taught as a foundational concept in biology.